Hey everybody, we're going to do a quick lecture today about biogeography, and this is chapter 18 in your textbook. So we're going to jump right in. Key concepts we're going to talk about are patterns of species diversity and distribution very globally, regionally, and locally. That global patterns of species diversity and composition are influenced by geographic area and isolation, evolutionary history, and global climate. And then regional differences in species diversity are influenced by area and distance, which determine the balance between immigration and extinction rates. So to start out with, sort of the idea of biogeography is why are species distributed as they are across the Earth? And so to give an example, 1 hectare of rainforest in the Amazon contains more plant species than all of Europe. And you may think, well, why is this? The Amazon basin is the largest watershed in the world. The number of fish species in the Amazon River exceeds the total number found in the entire Atlantic Ocean. And so you can think about the reasons for this, and there are multiple reasons that you can probably come up with, and some of these have been tested by scientists. So to talk a little bit further about this idea, we want to talk about global water distribution, or the distribution of water across the Earth. And you can see in the pie graph on the left of your screen here that the vast majority, 97.25% of all the water on Earth is distributed in the oceans, or is found in the oceans. So that leaves 2.75%, and so there's ice that makes up the majority of that. It's about 2%. Groundwater is 0.7%. And then all other water on Earth is only 0.05% of the total distribution of water. And so if we look at all other water, you can see that lakes make up the vast majority. And then rivers and soil moisture. And rivers is only 1%. I'm sorry, lakes is this this part here. No, we're right. I'm right. 60% are lakes and 33% soil moisture, and then rivers and are here. I can't actually remember what the small slice there is. But the reason I'm telling you this is because we want to take a look, where are all the fish found in the water, in all that water on Earth. So freshwater versus seawater. About 58% of all fish are found in the oceans. However, remember that the oceans make up almost or just over 97% of all the water, so you'd expect maybe more. And then 41% are found in freshwater, and then about 1% are diadromous, or fish that spend part of their lifetime, their life history in freshwater and part of it in saltwater. And so this is sort of the geographic distribution of freshwater fish. And you may think, well, why is it that that fish are distributed in this way? And a lot of it has to do with productivity. The vast majority of the oceans are not very productive because they're so far from land, and there's no nutrient input, and most of the nutrient input comes in through erosion and that type of thing that ends up in rivers, and eventually in oceans. And I also think that the ocean, or the freshwater rivers and streams, are highly diverse in terms of habitat, and so it has allowed a lot of speciation and diversity to develop. And of course, most of the diversity in oceans is found in coral reefs. So habitat diversity makes a pretty big difference. Maintaining biodiversity hotspots. When these ecosystems that are highly diverse get disturbed, there's a devastating species loss. Deforestation, for example, has resulted in extreme habitat fragmentation, and it makes it more difficult to maintain species diversity. We're going to talk more about the theory of island biogeography, but it explains why more species are found on large islands than on small islands. And these types of ideas can help us answer the important question: what is the minimum area needed to maintain species diversity? And this is one of the really big questions for conservation biology. And one of the leading scientists, Edward Wilson, E.O. Wilson, suggested that we need at least 50% of the Earth, by the Earth's surface, put towards species diversity and conservation. He has I think it's called the 50% Project. that you can check out. It's pretty interesting. So on to the introduction. Physical factors and species interactions are important regulators of species distributions at local scales. But global and regional scale processes are also important in determining distributions and diversity of species on Earth. So to define biogeography, this is the study of variation in species composition and diversity across geographic locations. So simply, where are species located on Earth, and what is the richness and diversity look like in these areas? So the Amazon rainforest is most species rich with about 1,300 tree species. The boreal forests of Canada have only about two species that cover vast areas. So you can see the difference in those two large geographic regions, and again start thinking about why that is. Species richness and composition vary with latitude. So low latitudes have more and different species than higher latitudes. So you can think again about the Amazon rainforests versus those boreal forests in Canada. From continent to continent, even where longitude and latitude is similar, there are quite a lot of variation in species richness and composition. And they also vary within the same biome type, so depending on its location on Earth. So you think about biomes being large areas that should be very similar, but because of their different location on Earth, they can vary quite a lot in richness and composition of their species. And we'll talk a little bit about that towards the end of the lecture here. So a number of, or I should say, now we're talking about biogeography and spatial scale. So a number of hypotheses have been proposed to explain biogeographic variation, which are highly dependent on spatial scale. So spatial scale is really important when it comes to biogeographic variation. Spatial scales are interconnected in this hierarchical way. We'll talk about a little bit about that here in a few minutes as well. Patterns of species diversity at one spatial scale set the conditions for patterns at smaller spatial scales. So if we look at this next slide, you can see this figure on the right sort of has this effect where you look at an area in North America, and looks like it's probably supposed to be around the Appalachians. And you can see that there are these mountains. If you look down in this area, we have where the arrow is coming from. And look at in a large part of that, you see we have this sort of looks like some kind of aquatic system. And then the forest here. And then if we look closer in just this area of the forest, you can see that there are coniferous trees. And so this is linking these global, regional, landscape, and local scales. And so if we take a look at what this means, so regional scale are areas with uniform climate. Species are tied to that region by dispersal limitations. So examples are the Amazon and the Canadian boreal forests. So species can't really leave that area because of the dispersal limitations. So an organism in the Amazon can't really make its way all the way to the boreal forests of Canada and vice versa. Regional species pool, these are all the species that are contained within a region. This is called gamma diversity. Landscape scale is determined by topographic and environmental features. Okay, so you can see again here, this is the landscape scale. And local scale. Local scale is equivalent to a community. So at this scale here, in the bottom panel D of this figure on the right, that would be representative of a community. And species physiology and interactions with other species are important factors in the resulting species diversity. And this local scale diversity is called alpha diversity. And we're going to look at a comparison between alpha, beta, and gamma diversity. And beta diversity is the change of species or turnover from one community type to another. And this is what connects local and regional scales. So let's take a look at those three. So in this figure, you can see the site A, site B, and site C. Site A has these three different species, this black species of fish, this orange species of fish, and a yellow species of fish. Site B has orange species of fish, just like site A, as well as this sort of yellow color. But it also has this red color species fish. And then in site C, you'll see that there is this green species or yellow species, and then this black striped species. And so in site A, alpha diversity is 1, 2, 3 different species. Site B is the same, 1, 2, 3. But in site C, you can see that alpha diversity is 1, 2. Now, beta diversity, remember, is comparing between two sites. And so if we compare beta diversity between site A and site B, there are 3 species in site A, so you can see the 3 here, minus 2 because there are 2 species that are in common with site B. And then in site B, there are also 3 species and minus the 2 that are in common with site A. Okay, so that is 3 minus 2, which is 1, plus 3 minus 2, which is 1. So AB beta diversity is 2. And then if we compare the beta diversity between site B and site C, you see that there are 3 species in site B, only 1 in common, the yellow species with site C, and then there are 2 species in site C and only 1 in common with site B. And so the beta diversity between B and C is 3. And then gamma diversity, and gamma diversity is the diversity of the entire region. So if we have this black species, an orange species, and yellow species, that's 3, plus this red colored species, that's 4, plus this black striped species, that's 5. So the overall gamma diversity in the ecosystem is 5. So it's just a way to look at species diversity in more than one region or one site. So biogeography and spatial scale continued. The regional species pool provides raw material for local assemblages and sets theoretical upper limits on species diversity for communities. The relative influence of regional and local processes can be determined by plotting local versus regional species richness. So on the figure on the right, you see that we have local richness on the y-axis and regional richness on the x-axis. And so when we have this situation where there's a one-to-one relationship, regional and local species diversity are equal, all species in a region will be found in all communities. So in this larger area, it doesn't matter how big an area you sample, the local richness and the regional richness are equal. And then if local species richness is proportional to regional richness, the community richness is mostly determined by the regional species pool. So the regional species pool is mostly dictating this blue line. Okay, and you can see that that's because regional species richness increases at a greater rate than local richness. And then lastly, if local species richness levels off, then local processes can be assumed to limit local species diversity. And so you see this local, or this leveling off, this plateau of the red line down there. So now we can talk about a little bit about Alfred Russel Wallace, and I know a lot of you are interested in him and his contribution to natural selection, the theory of evolution through natural selection, and he's sort of more well known as the father of biogeography. He did certainly contribute, along with Darwin as the co-discoverer of the principles of natural selection. But his major contribution is really this idea of the study of species distributions across these large spatial scales. And he noted that mammals in the Philippines were more similar to those in Africa, which was 5,500 kilometers away, than they were to those in New Guinea, which was only 750 kilometers away. And this, which obviously seems illogical. It seems like the closer to land bodies are, then the more similar the species should be. So this really led him to think about these two, the demarcation between these two faunas, and this further became known as Wallace's line eventually. I think Huxley was the first to call it Wallace's line around in the late 1850s. But Wallace published this geographical distribution of animals in the year 1876. And he overlaid species distributions in geographic regions and revealed these two global patterns. One was that land masses can be divided into 6 biogeographic regions, and you can see those over on the panel or the figure on your right. And also there's a gradient of species diversity with latitude, so higher diversity at the equator, and then as you move to higher latitudes, you get less and less species diversity. And also these 6 biogeographic regions correspond roughly to the Earth's major tectonic plates. And so this is driven largely by continental drift. So you can imagine that the flora and fauna on continents should be somewhat similar because of the long time period that has been allowed for the for species to evolve. And then the legacy of continental drift can be found in the fossil record and in existing taxonomic groups. And then there's this term called vicariance, and vicariance is the evolutionary separation of species by barriers such as those formed by continental drift. And then tracing the threads of vicariance through provided evidence for early theories of evolution. And as Wallace was amassing all this information on species and he made these geographic connections among them, his ideas about the origin of species started to solidify, and that's sort of how he was led to the idea of natural selection that he shared with Darwin, and then they co-produced a paper or a reading in the Royal Society of London. And then contemporary research continues to expand on Wallace's biogeographic regions, and your textbook goes into details about that. I'm not going to spend time here talking about that. Onto global biogeography. Biodiversity hotspots are those areas of high species richness that are under threat from human activity. This is sort of a hot topic, no pun intended, and back several years ago. Still important for sure, and there are certain areas that we feel, scientists feel, are really important for conservation and to conserve and keep protected, because of that, the reason that they can provide evolutionary diversity moving forward as climate change continues to, and natural extinction rates continue to move through time. And global patterns of species richness are ultimately controlled by rates of speciation, extinction, and dispersal. Assuming similar dispersal rates, species richness should reflect a balance between extinction and speciation. Speciation rate minus the extinction rate gives the rate of species diversification, and this is the net increase or decrease in species over time. What ultimately controls this rate? There have been many hypotheses proposed, but there's little agreement on this. There are multiple confounding latitudinal gradients of area, evolutionary age, and climate correlated with diversity gradients. So there's quite a lot of hypotheses and a lot of discussion, I guess you'd say, about whether or not what or which of these are most important and when, where, and when. And then manipulative experiments are, of course, impossible because of the global scale. Species diversification rate is the tropics have the most land area on Earth, and temperatures are very stable, so they tend to have the highest diversification rate. Species, that should be a 2 there, species diversification time, so the tropics are thought to have been more climatically stable over time, and species therefore have had more time to evolve. And then thirdly, the productivity, so the terrestrial productivity is highest in the tropics, and so all of these things lead to the tropics being seen as the cradle of diversity. And loss of biodiversity in the tropics could cut off the supply of new species in higher latitudes in the future. So we know that extinction is a natural process. However, the current rate of extinction is way greater than it has been at any time in history that we know of, as we're basically in another mass extinction event. And so if we continue along this path and we lose biodiversity in the tropics, then it could really reduce the ability for life to sort of expand and evolve new species to occupy these unoccupied niches further from the tropics as time goes on. Regional biodiversity continue. This an important concept in biogeography is the species area relationship, and this is basically that species richness increases with sampled area. And that may seem somewhat logical, but you could also think that once you get to a certain area sampled, you may end up with all of the species. So no matter how many more sites you sample, you won't get any more species. That relates back to that earlier figure where we talked about the one-to-one relationship. And then species area data are not usually linear, and so S and A in this figure down below are log transformed to obtain a straight line. So this is the unlog transformed data, so this is just an arithmetic axis, so the number of species on the y-axis and area on the x-axis, and you can see that there's a rapid increase the greater area that you sample and the number of species, and so eventually though it starts to plateau. So you sample larger and larger and larger areas, and you get less return in terms of number of species, new species that you accumulate. So if you log transform that, you get this linear line, or that sounds funny, but a straight line. And so let's see. This is the equation. Species richness is equal to the z, or slope, times the area plus c, or the y-intercept. And so it can allow you to calculate, depending on how large of an area you need to sample, to calculate the species within an area, or to capture all the species in the area. So talking now more about sort of getting into island biogeography a little bit, and an island can be any kind of isolated area surrounded by dissimilar habitat, so or a matrix habitat. And so even things that wouldn't normally you wouldn't normally think of as islands, so mountaintops, for example, are sometimes considered to be islands, particularly for certain types of species. So and we also know that all these islands basically display the same basic pattern, and that's that large islands have more species than small islands. So for example, if an Arctic community that has of a really tall mountain would have more area. So if you think of it as an island where there's some lower limit that would represent sort of the water, if you will, it may not be water, maybe a tree line or some thermal tolerance, but the bigger, the higher the mountain, the more area there would be, and so the more species you would expect to be on that island. And the same holds true with traditional islands in for example, the ocean. So island species diversity also shows a strong negative relationship to distance from source of species. In other words, the farther you are, if you're an island, the farther you are from the mainland or unfragmented habitat, the lower your species diversity. And in 1963, MacArthur and Wilson, and this is E.O. Wilson that I was talking about earlier, plotted bird species richness in the island area for a group of islands off of New Guinea. And the islands of equal size had more species if they were closer to New Guinea. And so they developed, MacArthur and Wilson developed their theoretical model. It's called the equilibrium theory of island biogeography, and basically stated that the number of species on an island depends on the balance between immigration or dispersal rates and extinction rates. And so we'll take a look at this figure in a second, but if immigration and extinction rates are plotted, the number of species on the island should fall where the two curves intersect. The equilibrium number is the number of species that should theoretically, quote, fit on the island, irrespective of the turnover or replacement of one species with another. And so if you look at this figure on the right, you can see the rate of immigration or extinction, immigration being the red lines and extinction being the blue lines, and then the number of species present on the x-axis. And so the islands that are far from the mainland have a low immigration rate. Islands that are near a mainland have a greater immigration rate. And then islands that are small have a greater extinction rate than those that are large. And so if you look at a large island near a mainland, you can see that this would be the expected number of species. And as you can imagine, as a smaller island, but still near, you'd have fewer species. Sorry, that's my dog, or one of them. Pearl, come here. We have coyotes that come by our house, so sometimes she gets a little rambunctious. Anyway, assumptions of this model or this theory are the island size controls extinction rates. Populations on small islands have higher chance of going extinct due to their small population size and increased effects of competition and predation. And also that distance from the mainland controls immigration rates. So distant islands should have lower rates than near islands. Your book goes into more detail about these and also some follow-up experiments, and I'm not going to talk about those in the discussion here. And then in 1969, Simberloff and Wilson manipulated mangrove islands. This is just the craziest thing. Such a cool experiment, but also just crazy. I don't think you could get away with this at all today. But basically, they sprayed these mangrove islands with insecticides, and they killed all the insects and spiders. And then they went back a year later and evaluated the number of species, and they found that the species numbers were similar to numbers found before the experiment, and also that islands closest to the source of colonizers had the most species. So you can see this in the figure over here, where the number of species, and the blue is a near island and the red is a far island. So the number of species increased more rapidly. This is days since defaunation, since they killed all the plants, and this is the number of species. And so the near island increased more rapidly and also had a higher number of species than the far island. And so the question is, of course, does the equilibrium theory apply to mainland areas? And mainland areas tend to have a higher rate of immigration because there are fewer barriers to dispersal. So it's easier for organisms to make their way into a decimated habitat on mainland because they can travel there by foot than it would be for animals to make their way over a large body of water, for example. And extinction rates are also lower because of continental immigration. Species on mainlands will always have a good chance of being, quote, rescued from local extinction by other populations. And the result is a less steep slope for species area curves on mainlands. And so on the figure here on the right, you can see the Channel Islands off the coast of California has a pretty steep slope close to California, and then mainland France has a less steep slope in terms of plant species relative to the area. And with that, we'll end, and I look forward to seeing you guys again here soon. I don't think I'm going to have an activity with this exercise, but there will be a quiz most likely, so look for that. Take care.